I was muted. Okay. So one additional reminder, your question about review reminded me. Just remember that Brooklyn has SI tomorrow morning, I believe, from 8 to 10 in the morning. So if you want to get a jump on studying, Brooklyn is great. She's really good at this stuff. So if you're feeling a bit worried about the exam, I recommend that you go meet with her. Okay. So picking up with the end of cellular respiration. So right now we're talking about aerobic cellular respiration, so the kind that requires oxygen. This is where the oxygen is actually going to come into play when we talk about the electron transport chain and ATP synthesis, otherwise known as oxidative phosphorylation. Okay. So quick review about the mitochondria. We've been thinking about them, but we haven't talked over them together, at least discretely in class yet. So remember, when we're looking at a mitochondria, we have an outer membrane. We have, red isn't showing up too well. We have an inner membrane, right? The inner membrane is the loopy one. Those are our cristae, technical term, but we can just remember that there's an inner membrane and it has a lot of surface area. And there's a space between them. This space between the inner and outer membrane is called the intermembrane space. So inter, like between, membrane between these two membranes, and it's a space. So this is a space that's going to be really important as we think about the electron transport chain and ATP synthesis here in this final step. When we were talking on Monday, we were in the matrix, right? So we were talking about pyruvate being shuttled into the matrix, and then the TCA cycle happening here as well. So some of the stuff we're talking about is going to be happening in the matrix as well. Remember, in addition, glycolysis, right, was happening outside the mitochondria. So glycolysis, we don't even need mitochondria to do. Everything else we need to get into the mitochondria. So technically, we can either think of this last chunk of metabolism as either one big kind of umbrella step or two little steps. So technically, it's called oxidative phosphorylation. So oxidative, we're going to expect oxidation to be happening, right? So what we're really going to be oxidizing here, right, is all those reduced electron carriers. Now we're really going to be using them, so that we're going to be oxidizing them back to their original form, that NAD+. So our two parts here, which we can think of either as their own steps or like part 4A and part 4B, are the electron transport chain and ATP synthesis. It's the electron transport chain that requires oxygen. Someone joining us. Okay. So when we're looking at what's happening here, got a lot of words on the side, but pictures are really the easiest way to understand what's happening. So what's happening at the beginning is we have a bunch of NADH from all our previous steps coming here, right? We remember that we made NADH during glycolysis. We made some NADH during pyruvate oxidation. We made a ton of NADH in the TCA cycle, right? That was a big point of the TCA cycle. So it's all going here to the electron transport chain. Now, what happens with that NADH is we have a series of proteins in the inner mitochondrial membrane, so that loopy one, the squiggly one on the inside, right? And what they do is they're basically doing a series of redox reactions where we take off this hydrogen, we use that electron to that we're removing to kind of power this, and we shuttle hydrogens through and into this intermembrane space, so between the outer and the inner membrane. So we're building up a concentration of hydrogen ions here. Now this is active, it requires energy, And the energy is actually coming from the electron in that electron carrier. So we don't see ATP being used here. It's a different form of energy that we're using, and it's kind of specialized. Okay, so we're building up this hydrogen gradient. We also see that our second type of electron carrier is involved as well. So FADH2 gets oxidized to FAD. And again, this is powering movement of hydrogen ions, building them up, building them up, building them up, building a high concentration here, right? High concentration of H+. So this keeps happening and happening and happening until we get to the end. Our final electron acceptor is oxygen. So our final electron acceptor isn't one of these proteins in the membrane. In the very final step, we send these electrons to oxygen. Oxygen joins with a couple of hydrogens, literally 2 of them, and we make water. So this is the only place we're actually using oxygen in all of oxidative phosphorylation and in all of aerobic cellular respiration as a whole, as a big, long process, right? We didn't see oxygen in glycolysis, didn't see it in pyruvate oxidation, didn't see it in the TCA cycle, only happening right here. But it's super important because if we don't have oxygen, we can't end the electron transport chain. So everything backs up, right? It means we can't regenerate NAD+, which means we can't do glycolysis, right? It basically means we can't start the beginning of this process if we don't have any oxygen to accept it at the end. So this is fatal if you don't have oxygen, and it's all because of this tiny little bit at the end of the electron transport chain where oxygen is our final electron acceptor. Okay. So what happens after the electron transport chain is we've created this state where we have a lot of hydrogen built up in this intermembrane space. And we've moved basically all of the hydrogen out of the matrix. So this would be our low hydrogen area. Okay. Now we know from when we talked before about gradients that molecules want to move from high to low. And this is true of our hydrogen ions. And because they're ions, because they're charged, they can't pass directly through a membrane. The inner mitochondrial membrane has basically all the same properties as a cellular membrane would have, right? It's made of phospholipids. It's kind of the same thing. So the hydrogen ions can't move until we put a protein there that allows them to pass through. So when we have enough concentration built up in between the intermembrane space up here and the mitochondrial matrix down here, when the difference in the hydrogen ion concentration is really big, we then allow this gradient to release, and we allow them all to flow through this protein we call ATP synthase. And ATP synthase, as these hydrogen ions flow through, does this kind of spinning action. And as it does this spinning action, it tacks an extra phosphate group onto ADP, creating ATP. And this is really effective for producing ATP. Like we saw glycolysis, we made 2 ATP. We saw the TCA cycle, again, right? We made 1 per cycle, so 2 per molecule of glucose. We are making a ton of ATP using ATP synthase, up to like 30-ish ballpark. It depends on what type of tissue, what type of organism you're talking about. So you don't need to remember a specific exact number, but it's an order of magnitude more than we create during glycolysis or the TCA cycle. So this is why this step is really important. So here we just see our hydrogen gradient illustrated again. All right, so we have our high hydrogen, our high hydrogen gradient up here, our low hydrogen gradient here, which creates this desire for them to move, basically. There are other forces that are created by this hydrogen gradient, like a difference in pH and a difference in electrical potential, which are not topics for this class, but they're really just other ways to describe the fact that we have a ton of hydrogen here in this intermembrane space, building up, creating like a force for movement. So it's also called the proton motive force because a hydrogen ion is technically kind of just a proton, basically. Okay. So here we see our ATP synthase. It's spinning round and round as we allow these hydrogens to flow through into the matrix. And here we see that at the same time, it's creating ATP. So none of this process here in the mitochondria requires ATP. We only create ATP here. We're not going to worry about this word, chemiosmosis, but that is the technical term for what we're doing and the way in which we're creating ATP here. So this is an overview of the whole process, just looking at where we are relative to the mitochondria again. So here we have our outer membrane in dark orange. Here we have our inner membrane in light orange. There's a space between them. We know glucose is converted into 2-pyruvate molecules out in the cytoplasm, creating some ATP and creating some NADH, right? We know that pyruvate moves all the way into the center of the mitochondria, into the mitochondrial matrix. It's converted to acetyl-CoA as it loses some carbon dioxide and also creates some more NADH. Acetyl-CoA goes into the TCA cycle, which is kind of constantly revolving because it's starting with that oxaloacetate that joins with acetyl-CoA and it's regenerating it at the end of the cycle. The TCA cycle gets rid of all the remaining carbon from our original glucose molecule. So that's given off as carbon dioxide, a waste product. Creates a tiny bit of ATP, but the big thing it's doing is creating a bunch of NADH here. Also creates our second electron carrier, FADH2. These all go to the electron transport chain. So the electron transport chain is physically embedded in the inner membrane, right? The chain is really this series of proteins. And its job is to pump all these hydrogen ions into the intermembrane space, right? So here's that space. Here's that space, right? It would continue this way until we have a big concentration. We allow it to be released through the ATP synthase molecule, creating a bunch of ATP. So this is really all our steps that are part of aerobic cellular respiration. So we have glycolysis, pyruvate oxidation, the TCA cycle, the electron transport chain, and then at the end we have our actual ATP synthesis where we make a ton of ATP. So our overall equations here, if we think about where we were at the end of the TCA cycle, right, at the end of the TCA cycle, we'd put in 1 glucose molecule. We put in 10 molecules of NAD+, right, because we had to put some in glycolysis. We had to put some in during pyruvate oxidation, and we had to put in a bunch during the TCA cycle. We put in 2 molecules of FADH, which is the oxidized form of FADH2. We'd gotten out 6 CO2, right? So we gotten rid of, glucose is a 6-carbon molecule, so that's actually all of the carbon from glucose. We'd produced 10 molecules of NADH, which is our most important electron carrier, 2 of FADH2, which is our other reduced electron carrier, 4 molecules of ATP, 2 of which came from glycolysis and 2 from the TCA cycle. When we do the electron transport chain, we're converting NADH back to NAD+. So we're going to cross out the NAD+ and the NADH because we're transferring them back. Same thing for FADH and FADH2. So we still have glucose here, but now we're going to add the things we need for, and we still have carbon dioxide here. Still have the 4 ATP here. So now we're adding the parts that we need for the electron transport chain, which is that we need some oxygen, our 6 O2 molecules, in order to be those final electron acceptors at the end of the electron transport chain. And when they accept those electrons, they join with some hydrogen, giving us water at the end of this process. So then we got one more bit to go until we have our full equation for aerobic cellular respiration. Where's my annotation tool? Okay. So this is just what we had above. So this is the state we are when we've built up that hydrogen ion gradient in the intermembrane space. Now, when we release that hydrogen ion gradient, allow the hydrogen ions to move through ATP synthase, what we have happen, right? This is all the same as above. So we're making a ton of ATP, right? And this tilde is because it's not an exact number, right? So I said about 30. 30 plus 4 would be 34. If we made even more, we got even more ATP. The point is just it's a lot of ATP that we're making here. Okay. So that's it for me for right now. So our problem set looks like this. You're going to label what's happening here as the electron transport chain is running. So you're basically just going to be comparing to these pictures that we have in our slides and transferring them onto this slightly different picture that's showing the same things. And then you're going to answer a couple questions, right? So you're going to tell me, were these molecules oxidized or reduced as we're producing that NAD plus and the FADH again at the end? We want to think about where they're going, like physically, where did we need NAD plus, right? So we can think about what we might be doing with that NAD plus that we're regenerating. And then we're just going to flip back, make sure we know where the electron transport chain was, think back where those channel proteins are, so why we might need them. This one, I'm going to just tell you the part of the electro. The reason that's in there is because the hydrogen ions have a charge on them. So think about what that would mean here. So we know what a chemical gradient is. So just describe a chemical gradient and say that it involves, but it also involves an ion specifically. I'm kind of telling you the answer here. And then you're just going to talk about where the protons are coming from, where they're going, what they're passing through, and why we care. Like what is this doing for us? Okay. So that should be it.
It didn't give me an option to join a breakout room.
Oh, okay.
I think it's because I got like kicked out earlier because my Wi-Fi stopped working.
Oh, I'm sorry. Okay, let me, you're in group 4, is that right?
With like Ariana and Derek and Taylor, yeah.
Yeah, awesome.
No. Okay, thank you.
Mm-hmm.
Sorry, I stayed because I had a question, but then I forgot it. So I'll leave.
Okay, if you think of it, let me know.
Okay, sounds good.